last week, we kind of introduced things, and then, we're looking at some of the features that bacteria have, and we talked about endospore formation, which is where we kind of stopped. And so part of that was just to remind ourselves what can happen, where we have bacteria that are endospore formers, and once the spores are formed, everything that the cell will need in its vegetative, part of the life cycle is contained within the spore. And so when we look at the various ways to prevent bacterial growth, and we talk about aspects that are directed at the spore, one of the things that we're going to be talking about there is that we want to be able to eliminate the spore, we want to be able to kill the spore, so that we have no chance of any vegetative growth. Okay, so we wanted to make that point. And so we had also looked at some of the features that bacteria have in terms of their transport and everything, the ways in which they can get nutrients in and out. And we were making a distinction between gram-positive and gram-negative cells. So I think by now, as way of review, we all remember that gram-negative cells have an outer membrane. Everybody remembers that? And so not only do we transport across the plasma membrane of the bacteria, but we have to also get through the periplasmic space, and we have to get things out through the outer membrane. And as well, if we're importing things into the cell, we're coming across the outer membrane, we're also coming across that periplasmic space, and then we're going across the plasma membrane of the cell. And so the outer membrane has a lot of channels and pores and various structures to allow things to move in and out. In fact, more than we have on the plasma membrane. And we had talked about the different transporters. I just wanted to make sure that we got that point. We talked about active transport, facilitated diffusion, regular diffusion, and we mentioned we have both primary and secondary active transport. We talked about the ABC transporters, we used an example of the primary transporters. We talked about the two binding domains that we have for ATP on the cytoplasmic side of the membrane, and so this is pretty much what we're seeing here again. I just wanted to make sure that we kind of remember those things. So again, we have the solute that's being transported. It gets to the ATP binding molecule. It binds on the outside. We have a conformational change that allows ATP to bind. So we have those nucleotide binding domains. And then so once that happens, the energy generated by having hydrolysis will allow the sodium to be transported in. And then we change conformation again so that we can repeat the process all over again. So this is just to remind ourselves of that. And so we're looking at again, primary active transport with ATP, the example being the ATP a binding cassette over here. And so we have it in bacteria, we have it in archaea, we have it in eukaryotes. So this is an important way that we can have things happen. We also have secondary active transport, which we talked about. And so again, we have the ability to transport things, in one direction. Uniport systems, we can transport two molecules at the same time in symport, or we can transport in the opposite direction with antiport. And so we just wanted to remember those differences, okay, so that we can do primary active transport, secondary active transport can also take place. And then we also mentioned group translocation, okay, as well. And so again, here we can have an interdependent, transport that we can have a modification. We mentioned, being able to add phosphate groups, right? And so one of the best studied type of group translocation is, with the phosphoryl pyruvate. So you're looking at metabolic pathways, and you see, the transport, of these sugars. We have an example there. So here we have the sugar phosphotransferase system as an example of group translocation. So typically we just want to remember the types. So we can do diffusion, we can do facilitated diffusion with carrier proteins, we can do active transport, primary, secondary, we can do group translocation across the membrane. And we have other things that the cells will do. So iron is important for a lot of organisms. And so, they need this, when we look at the metabolic pathways, and we're looking at electron transport systems and so on, we have the involvement of iron there. So we have to be able to take it up. And so, amongst different organisms, they have these siderophores that will actually aid the process, of getting iron in. So the siderophores will bind, to the iron, in this case, ferric ions, and then the whole thing is taken up into the cell. So, in this section over here, we just want to remember these features that the cells will perform. Now, we talked about LPS. And, when we talked about LPS, we said this was a major component of, was it gram-positive or gram-negative? Gram-negative cells, okay? And so we talked about, the parts of LPS. So we want to remember that. So again, we have, the portion that sticks out, okay? We have the O antigen, and we said that the O antigen was important for typing the organisms, okay? So we have antigenic differences in there. Antibodies made against them, can be used to type, for these organisms. And we mentioned, the core, if you have, in the middle, and then, the core polysaccharide, and then we have the lipid A, okay, which is the one that actually imparts the properties to the endotoxin. Okay, so we want to remember, those three parts. And so it's an important molecule because, as we saw last time, when gram-negative cells die and they shed this stuff out, it elicits all kinds of reactions in the body. Everything, from inflammatory type responses, complement, it has mitogenic properties, especially for B cells, and so on. So it's a very important molecule. And so, we see some of the things listed over here. We mentioned some, and these are additional ones. So, the negative charge on cell surface, we have with those sugars, from the LPS, the outer membrane structure is essentially stabilized. We can also use it to attach to things. We have a barrier that's generated, because again, remember, we have that outer membrane, and then we have the LPS, on top of that, kind of embedded, into the outer membrane. And so, again, the O antigen, is the protective device, for host defenses, and then the endotoxin component, the lipid A. And so things that we want to keep in mind. So that the negative, the gram-negative cells with the outer membrane, again, a lot of pores and transporters. And so the porins, you've seen a lot of mention of porin proteins. They're different, different ones, different types. These are found, in that outer membrane. And so, they would form channels to that, molecules, come into and out of the cell. And then there are also transporter proteins that can also be found in that outer membrane. So compared to gram-positive cells, so gram-positive bacteria do not have this kind of makeup, right? So, we have more exotoxin production, right, with the gram-positive cells than we do with the gram-negative cells. Okay, we don't have the endotoxin in gram-positive bacteria. Okay, so this is going to be important as we're looking at the list of bacteria, gram-positive, gram-negative, that we find in the oral cavity. Right, we want to be remembering these properties that we have between the two groups. Okay, all right. So, so the things that we want to kind of do, newly for today. And so we're beginning to look at this oral ecology, trying to understand, where we have the bacteria, what kind they are, what, and what kind of things they're participating. So we want to begin to look at this biofilm formation. We'll say a lot more about that tomorrow. But, we want to see some of the representative organisms that we find in this place. And then we want to look at the gram-positive, gram-negative cells that we have in the oral cavity, what kind are they, and the type of structures that they produce, adhesins that they use to adhere to surfaces and so on, and the differences between the exotoxins and the endotoxin, and we just talked about the lipid A, and, you know, what kind of processes, you know, disease processes do these bacteria, you know, get involved with. So again, you know, those 4 phyla that we mentioned, right? So we're going to see distributions of these different bacteria, you know, in these groups. And so we're going to look at, you know, the things that are associated with the development of caries, periodontal disease, endodontic diseases, the kind of organisms that are associated with that. And then in the next, you know, couple of meetings, we're going to look in a bit more detail as, you know, as to what's going on. So, you know, we have a list of organisms over here, basically, you know, kind of represented, and this is by no means a complete list. But we have, you know, an example of the distribution of the organisms that we find. So you see we have them, you know, listed as gram-positive, gram-negative. And so there are some key organisms already that we've been mentioning. So we have the Streptococcus, you know, a genus, you know, mutants being one that we've mentioned a few times. So we want to make sure we remember that one. Okay, all the way down to the Lactobacillus species and so on that we have, you know, amongst the gram-positive. On the gram-negative side, right, so we've mentioned Papillomonas gingivalis. You know, we talked about that one being potentially pathogenic. And, you know, besides that one, we have, you know, like the Fusobacterium species, etc. That, you know, Trichonema we already also mentioned, all the way down to, you know, the list that we have, the 2 different species of Trichonema. So again, you know, we have this large distribution of organisms. So we have, amongst the group, we have those that are symbionts, okay, that normally are not disease causing. And then we have ones, you know, that are opportunistic, as we talked about, you know, last week. And then we have those that are truly pathogenic, okay? So in the biofilms, you know, we can have, you know, the symbionts, you know, mixed in with the pathobionts, as well as with those organisms that are opportunistic in nature. So again, in the list, you know, we begin to recognize, you know, some of the organisms that are not commonly mentioned all the time, but so as of today, for example, I think, you know, we should all kind of appreciate the Streptococcus mutans. We're going to see that we have other species, you know, all the way to the Lactobacillus, you know, organisms. And then on the gram-negative side, we want to remember Papillomonas gingivalis, and then also Trichonema denticola down here, okay, that we've already mentioned before. And so what this is showing us, and this is, you know, from the article by Takahashi, and, you know, in the notes, you know, you have the journals, you know, the article references, the name, so that if you wanted to look further for yourself, you know, you can get access to those papers and, you know, kind of read the study to get some more information. But pretty much, you know, what is being shown over here is that we have a lot of work that's being done, you know, these last few years, you know, quite intensely. People have been very interested in trying to find out, you know, what the key organisms are that you find in various niches, you know, within the oral cavity. And whether you have the organisms acting alone in terms of, you know, the damage that they cause, or if they're working, interacting with other organisms. And so the consensus is getting stronger and stronger that we're not dealing with individual organisms, you know, responsible for a lot of the pathogenesis that occurs, but that you have organisms working together. Amongst bacteria, you have, you know, bacteria working together, and as well, in some situations, you have the bacteria also working with things like Candida, and, you know, to have the type of pathogenesis that progresses. And so the interest, you know, that he has is to say, okay, you know, what is the relationship between these organisms and what happens? So what you notice over here is that, you know, you have the organisms, you know, of these conditions that are important, you know, during caries, in periodontal disease, and, you know, the different organisms, we begin to get to more systemic complications, you know, with, you know, from the organisms. you have a different group of organisms that are involved. And so part of, what this is, and again, we're not memorizing this, we just want to kind of have illustration, is that, we see that within the mouth, the nutrient supply that's available from the foods that we eat, whether it's proteins, carbohydrates, lipids, all of these, contribute to what the bacteria are able to use and the byproducts that they have. Secondly, the proteins and other molecules that are found in the secretions, like in saliva and so on, also play a large part in terms of how bacteria are able to utilize these things for metabolism and the products that they now generate that can affect the oral cavity. And so, what you see is that, again, depending on what's being provided from them, right, you can get, everything from acids, you can get short-chain fatty acids that are produced, from the lipids. You can have environments that are at once highly acidic and all the way range to more alkaline environments. And as well, you can have a variety of products that are generated, from things like urea, from thiocyanates, you get hypocyanates that are generated, nitrates and so on, alcohols, and all of this within the environment. And so part of what now needs to happen is to understand what creates that environment that allows for these products to be made, and how does that now affect the way in which the organisms affect the tissue in the mouth. And so another thing that is discussed in this article is that, again, if you look at where we find the organisms, we have those that are above the gingiva. So we have organisms that are on the tooth itself. We have organisms that are on the mucosa in the cheeks. And then we have those organisms that are subgingival. And each different location has particular characteristics that are associated with those locations. And so again, if you see what happens as you look, right, we can have, within the environment, we have changes that occur that allow for a lot of, movement of molecules across the membranes of these cells. We have, other things that would happen, where you have in protein-rich areas, you can have things, move all the way to highly active environments. And within the organisms, if they have virulence factors, you can have more invasiveness in what happens in the tissue, and so on. So you have a difference within these environments, depending on what organisms you have there and what metabolic products they're producing. So another review from which we're using images from is the review by Cosalonga and Herzberg, and they have, again, they're addressing the same problem, in terms of the types of organisms, what happens if you have either less diverse or more diverse organisms, are we in a healthy state, are we in a state that we can classify as being a more diseased state. And so they go through and they classify, different aspects of the oral cavity and the various niches where you would expect to find, find bacteria. So as you can see, in this image over here, fine surface lettering, we have, the mucosal surfaces, including the gingiva. We have the surface of the teeth, crown, etc. We have the proximal spaces, between the teeth. And then on the surface of the teeth, we have, these pits and fissures. And in any of these locations, we have, biofilm formation with different groups of organisms that are represented in them. And as well, as we mentioned the cheeks, we also mentioned the tongue, sublingually under the tongue. We have these communities of organisms. And so the question is, are there ones that are typical that we would expect to always find in these locations? And are there, transient organisms that also can be found, within the cavity? And so we have the teeth, saliva, we have the dorsolateral areas, of the tongue surfaces. We have, the various, the gingival areas, sulcus, parietal pockets. We have the epithelial surfaces of the mucosa. And on the tooth itself, where we have, the surfaces. And so the changes that occur as a result of the bacteria metabolizing and producing acids, right, is what we have now associated, with caries. And so in this particular slide over here, we want to kind of remember these areas, and I think they're fairly straightforward in terms of remembering what they are. And so we have the illustration shown over here, and the major point that's being made is that the more diverse the organism type is, so if we have more diverse bacteria, right, we have more of a disease state. The less diverse we have of the organisms, the more we have a healthy state. And so a couple of terms that we need to keep in mind. Okay, so we already mentioned the terms, pathobionts, symbionts, and so on. But another term that we want to kind of keep track of are the keystone pathogens. Okay, so we have the organisms, that essentially, their presence, they will signify, pathogenesis, down the road. And so again, if you look in these two different sections, here we associate periodontal disease, right, with, a larger pocket that we can find, more pathobionts, associated with the symbionts, and then we also have the keystone pathogens that are present. And so again, we have the high diversity. In this section over here, we associate with periodontal health. We have low diversity, low richness. The pathobiont population, is very low. We have more of the symbionts, and again, very low representation of any keystone pathogens that are in here. And so, these two, the shift between these two is what, would indicate, the development of a pathogenesis. And the surface of the teeth, again, if we have, if we were to look at the environment, right, we have low diversity and richness on the actual surface, of the tooth enamel. But then within the pits and the fissures, we can collect bacteria there, and those numbers can, become quite diverse. And again, those we would associate now because of metabolic activity and the acid production. Right, we have this aciduric, organisms, in here. Then again, we can associate that more with being able to have caries. So we have, a few images just to kind of look at, and we're not going to belabor this, just to show the thing. So again, you can see the differences, between what happens, in the healthy situation versus, what we have now, with the presence of something like Porphyromonas gingivalis, Prevotella, Tannerella, and we can see, the inflammation, that's occurring as a result of these. So we need to remember these organisms. We're going to see them over and over and over again. So, and again, we find them associated within these environments. So again, we can tell the difference between the healthy and the unhealthy situation according to the number of organisms that we have present. Things can get pretty severe, and we have some other organisms that are associated with this. So the Actinobacillus genus, was changed recently to Agrigobacter, so we want to remember that change. And so when we see, aggressive, in this particular condition, aggressive juvenile periodontitis, this organism is most associated with this condition. So again, we need to remember the different types of bacteria that we're finding. So again, you can see, how severe this looks in terms of what's going on in the tissues. So if we look at the environment, and so again, we're seeing, when we look at the types of plaque, the location that we find. So if we have, saliva coating the teeth, so the supragingival, right? We can find, that we have carbohydrates, we have, proteins and things from the saliva. We can get anywhere from neutral to highly acidic, and most of these organisms, we can find aerobic organisms in there. And again, these are saccharolytic, so they're dependent on the sugars, for metabolism. Subgingivally, again, we're looking at, the location, within the gingiva. We are beginning to affect the epithelium, the mucosa. And again here, with the alkaline, oxygen levels, we're dealing with anaerobic organisms. And so again, these are proteolytic, amino acid degrading. And then if we compare that to what we have, on the tongue, we have again, organisms that can be found there. So we have the saliva-coated epithelia. Again, we're looking at carbohydrates. You can get from neutral to acidic, and we can have aerobic organisms here. And so we have a mixture of organisms that depend on sugars as well as on proteins. And so the main point of this is to remember the environment. Okay, because when we talk about the biofilms, what we're going to try to keep in mind is that in the core of the biofilm, we have, a lot of, anaerobic organisms. As we work our way out, we're going to begin to find some facultative organisms, right? And then we're going to find the aerobic ones, kind of the periphery. And so this makes a difference in terms of what happens within that biofilm because of this kind of arrangement that we have with the organisms. And so again, you want to remember those things. Okay, so again, we're looking, at similar to what we've already seen in the first two images, and just showing, that we can have, above the gingiva, these are supragingival calculus, and again, organisms can be identified from those. And, we can say the things we're saying about the types of organisms because if you, were to stain that material, you can identify, the gram-positive, gram-negative organisms, and you can speciate in some cases, which organisms they are. And so again, ultrastructurally, you can see the same thing as we're seeing on the top figure. So again, this is that list of organisms. So we've mentioned the Porphyromonas gingivalis, Trichonema denticola, Streptococcus mutans. So we want to remember these particular organisms. We want to know, again, if they're gram-positive, gram-negative, right, and the different characteristics that we talked about last week, okay, morphologically. Okay, we want to remember those things. So again, this is put here again just to remind to say that again. So we're looking at either the concord appearance of the distribution of organisms, when we have dental disease states. And so these, are done to pathogens, which is what they are, the ones that are going to cause pathogenesis, right, can form this type of plaques, look like a corn cob. So that's the characteristic of them. So when we see those, we know that we're dealing with these organisms that can form these type of plaques. And so the important thing here is that we have the acids being produced from metabolic activity, and then we also have the presence of virulence factors that allow them, to cause the, the distribution that we're seeing in the image. And so again, the different locations we want to be able to remember, and in this case, when we have the plaques form, depending on the location that we find the organisms, right, we can see this co-aggregation that occurs. And so remember what we just said at the beginning. So in the individual organisms not causing these plaques by themselves, we have interactions that are taking place. And so when we have all that acid that's generated, from the metabolism, we have that affect the enamel over a period of time, then we can begin to cause destruction, on that enamel and so on. And so the acid, formation is necessary to have the caries that now develops. And so the other thing that's important over here is that, the initial organisms, that establish the biofilm, right, very important in beginning the damage process going on, and then as other organisms come in to interact with them, then we have the plaque become even much larger and worse on the tooth itself. And so we have this coaggregation that occurs. The cells are able to adhere to each other, so they produce adherence molecules that allow them to attach to each other. And so we mentioned structures like fimbriae last time. We mentioned the hilli that are used for this process, but we also have proteins associated with this interaction. And so it's important to remember that aspect of things. So when we look at what happens as we have caries being formed, when we have these organisms present, we have a cycle that goes on. So we have a demineralization taking place, and then we remineralize. So we have this cycle going over and over again. So we produce the acids, we begin to damage, the tooth surface, and then as we're producing the saliva, right, we have, molecules in there, we have, proteins, we have ions, etc. We kind of, remineralize again. So we keep going through this cycle. And so, the longer this goes on, right, the worse it is for the tooth surface, and then we get the caries that results. Now, so this is different from what happens when we have periodontal disease. So here we don't have, an immune response going on, okay, recruiting cells, that are going to be part of the process. So we don't have neutrophils coming in and having granules being, releasing contents and so on as we would have when we have, periodontal disease. But the cycle is what's important, and it's, clearly a result of these organisms being present, okay, on the surface of that tooth. So in this particular case, keeping bacterial numbers down is pretty important. And so, there are no, vaccines or no drugs that are directed against this. The whole idea is, good dental hygiene. Again, keeping those number of organisms down. So last week, I mentioned that one of the things that goes on, with understanding the bacteria and other organisms that we have in the mouth is, trying to stay steps ahead of the increase in growth of these organisms. So we're pretty much, trying to prevent, damage from occurring by keeping the numbers of the bacteria down. And knowing those particular organisms that are involved becomes important because then we can target them. As we already said, we can have antimicrobial agents that are pretty broad that can go after, both gram-positive, gram-negative, but we have ones that are directed towards, gram-negative or towards gram-positive based on the characteristics that we have for those. So again, different from caries, with periodontal disease, we're looking at inflammatory type reactions. So the immune system is heavily involved, and we're recruiting cells to the area. So initially, the organisms will stimulate innate immune responses. So you're going to have toll-like receptors interacting with molecules on and from the bacteria. We're going to have inflammatory cytokines being generated. We're going to recruit neutrophils to the environment. We're going to have T-cell involvement as we progress with more adaptive immune kind of a response. And so together with the organisms and the immune response that's taking place, right, we can begin to actually affect the tooth itself and the support for that tooth. And so the initiation begins with the bacteria that are in the biofilms that we have in the subgingival plaque area, which now stimulates the immune response, and then together we're having this damage that takes place. And so Porphyromonas gingivalis has been associated with a lot of this process. So with the caries, right, they have Streptococcus mutans, and some of the other strep species. But when we begin to look at periodontal disease, Porphyromonas gingivalis is pretty important. And again, many times, Porphyromonas gingivalis is found with treponema, okay, so together with that. And so it's not just it by itself, although it is a keystone pathogen, and so there's a lot of interest in it. And if you look in the literature, there are volumes and volumes of just trying to understand this particular organism, okay, to find out how it can be prevented and managed. And so we have the key thing here. Besides, what we've already talked about with caries, the key thing is that this involves an immune response. So, part of the way your questions are going to be asked, would be that. And so we're going to look at the different types of periodontal disease, of which there are about 7 of them or so. And we're going to emphasize, the involvement of, different aspects of the immune response with that. But, you know, again, the thing here is we have an inflammatory response to the bacteria and the plaque. And so, you know, we have the swelling of the tissue, you know, affecting, you know, the pockets, and then we can get bone destruction, and so on. And so, for something like this, you know, even, you know, with the gingivitis situation, there is no vaccine or anything that's required for something like this. If things get, you know, pretty severe, you know, oral surgery can be done to clean things out. Antibiotics, you know, can be provided, and so antibiotics, again, we have broad spectrum, as well as ones that are, you know, geared towards gram-negative, gram-positive, right? So remember that cell wall business with the peptidoglycan that we talked about last week, right? So, you know, we have, you know, antibiotics that are directed towards that will pretty much be good for gram-positive cells. It will marginally affect gram-negative cells, but heavily against gram-positive. And so again, removing the plaque, you know, which means, you know, we're decreasing the number of bacteria present, and then, you know, good dental hygiene and that kind of a thing. So we want to kind of keep this in mind, you know, when we get to the point where we're going to talk about the different types of a periodontal disease. And so when we look at the types there, we're going to find that some of, you know, some types are associated with chronic conditions, you know, within the body. So, you know, things like, you know, type 2 diabetes associations and so on. And so we're going to look at that. So in the questions we have over here, which of the following organisms found in the oral cavity is gram-positive? Okay, which one? Streptococcus, right? Okay, so we want to kind of remember that, so that, again, that's a reminder to please look at the table. So, you know, we're going to need to know these organisms. I mean, you know, there are some of them that we will just mention in passing, but there are others that we'll mention over and over again, okay? So those ones we want to make sure that we remember. So what, you know, last week we talked about situations where, you know, of the 700 or so, you know, different types of organisms that we have represented, you know, a good 50% or more of those cannot be cultured. Okay, so we all remember that. So, but if we cannot culture them, and so we have no way of knowing, you know, what they look like, right? You know, how do we know those organisms are there? We talked about, you know, ways of doing that. How can we tell that the organisms are present if we cannot culture them? What can we do?
Blast the DNA and profile it.
Okay, right, so before we do the blasting, right, we can, you know, we can use PCR, right, polymerase chain reaction, we can amplify DNA, right, and then, you know, once we do that, you know, we can sequence that DNA, and then we can blast it, and then we can use that information, you know, to find out, you know, if the organisms we're finding belong to already known, you know, phyla, or if we have something totally new, right? And so, you know, so that's, you know, a good thing to be able to do that. And so, you know, if we find out that we have, you know, particular organisms represented, okay, between those that we can actually grow and show, you know, morphologically, you know, what they are, and as we're going to find out, you know, just a little bit, we can do metabolic, you know, tests, you know, with the fermentations and whatever, and find out what they are, okay? So if you suspect that you're dealing with, you know, organisms that are pathogenic, okay? So not every practice is going to be growing plates and, you know, doing that kind of a thing, right? But, you know, in some situations, you know, if something is pretty serious, there may be a need for it. And so what you're showing over here, what we're seeing over here, is the fact that, you know, from that suspected agent, right, we can culture, okay? We can try to get information, you know, from that, you know, morphology, we can gram stain, whatever, that's the most basic thing that we can do, right? You know, we can use microscopy, and so we have a whole range of things from light microscopy to immunofluorescence methods with confocal microscopy and so on, all the way to electron microscopy that we can do. You know, we have a variety of, you know, rapid tests, you know, immunoassays that can be performed based on antibody reactions. And so those include everything from Western blots to ELISAs and, you know, so on. And we can look for complement activity, you know, within the host And we can do molecular type, tests, which include the PCR that we just mentioned, and the sequencing and so on. And then we can also do a variety of biochemical tests, which includes, metabolic profiles of the organisms. And so when this is done, we get a better picture of what we're dealing with. And so in those cases where, again, we can't grow, we can still get, pertinent information that will allow us to know at least, what type of organism we're dealing with. And so we can identify things. So we have preliminary type of identification methods that are done. We can do things that are more definitive. And again, from that list that we just looked at, and they all include, direct ways of doing things right here. And then we can have indirect ways of doing things using antibodies, to probe. Either antibodies from individuals themselves, from the patients, or using, antibodies, to known molecules from, commercial situations to actually try to get some reactivity with, the antigens that we have on the organisms that we're isolating. So you need to know the difference between those things that are direct identification methods and the ones that are indirect type of methods. So the nature of the pathogen, is important. And again, as we already said, for some situations, culture-based techniques are just not available, or they're not going to help, in which case we need, we need the molecular techniques. And so in the next couple of slides, what we're going to be looking at, and we mentioned this last week, is that we have, these dichotomous keys. We have ways of going through to identify things. And, pretty much, every microbiology lab has some aspect of this available that's used. Everything from clinic, to research labs, to food industry, doesn't matter. Anywhere you work with bacteria, this can happen. And so in a limited way, okay, we're going to need to know a little bit of this. And so what you're looking at, so you start up at the top over there. If you have gram-positive bacteria, you want to know, okay, what shape they are. So do we have rods? Do we have cocci, right? And so based on, one or the other, there are additional questions that you're going to ask. So if we have cocci, we're wondering, okay, are we anaerobe? Okay, are we aerobe? Okay, so generally, keeping in mind that we can have facultative anaerobes and facultative, aerobes, right? And obligate aerobes, okay, and obligate anaerobes. Does that make sense? Okay, so we have that. But in any case, so if we have them and, we have that aerobic, okay, then we proceed down looking to see if catalase is present. If they're anaerobic, okay, we kind of stop there. There's somewhat other things that we can do with that. But if they're aerobic, we check for catalase. If there's no catalase, there's strep. If catalase is present, right, we want to see what kind of fermentation they have with glucose. And then, if we have, a fermentation taking place, then we have staphylococcus, and so on. If they're negative, micrococcus. And then to show if, these potentially pathogenic or opportunistic, right, then we see if they're producing coagulase. If they are, then, we're dealing with Staphylococcus aureus, because this can differentiate Staphylococcus aureus from Staphylococcus epidermidis. And so these dichotomous keys are pretty important that way. So for the straightforward things, since we have Staphylococcus, we have Streptococcus, we're going to be mentioning Bacillus, looking at Clostridium and so on. The basic differentiations on those, we're going to need to know. So coagulase production, catalase production, right? We begin to deal with organisms, that potentially could, invade and could actually, lead to a pathogenic state. And so other things, right? So if we look at the, fermentation, negative with the glucose, and we have strep, right, then we begin to look at other things, so, biosolubility, absorption sensitivity, etc. And then we can begin to break them down into things like strep pneumonia and so on. And then we begin to group them according to the Landsfield groupings into those that are group A, B, C, D, etc. and so on. And so all the way down to the bottom of the key right here, right? So the major groups, group A, D, etc., B, and then we have all the other Landsfield groupings. And so this is a good point, to say that the Landsfield grouping system, okay, was one that was, based on serology to identify, these different organisms based on the way we have antibodies, reacting to these cells with that, peptidoglycan that they have. So when we see, that Landsfield grouping, we need to be able to associate that with serological identification of serotypes, okay, of the streptococci. Okay, so that's a very major thing, that we need to keep track of. And then on the other side, so if we have, bacillus, then, we want to find out are they acid fast. So remember, we can use Gram staining to differentiate the bacteria, right? But not every bacteria is going to, react properly with the Gram stain. So we have Gram variable organisms, and so if we have Gram variability, we want to find out if they are acid fast or not. Okay, so the acid fast organisms were associated with things like mycobacteria, right? And so if we do the acid fast, if they're negative, then we want to find out, okay, we have endospores in here, we're going to go that direction. If, we find out they're acid fast, then we want to, say, okay, we're probably dealing with mycobacteria, okay, the most common ones. Okay, so if we have the endospores, we want to find out, again, if they're there, are they aerobic or are they anaerobic? Because we want to differentiate, things like Clostridium from Bacillus, right? And so again, when we do that, we want to find out, if these things, are motile or not. Okay, do they have flagella or no flagella, and so on. And so if we, have the endospores, we're aerobic, right? If we're anaerobic with Clostridium, if we're aerobic with Bacillus. And then on this side, if we don't have the endospores and looking at motility, then, again, we want to see are we aerobic or not, and then we can further differentiate things all the way down to, again, catalase positivity and be able to get, things all the way to Corynebacterium. So the basic differentiations on the key, okay, you will be responsible to know. So coagulase, catalase, acid fastness, okay, those basic things to differentiate. So these keys, kind of help with those type of things. Okay, on the other side, we have, with the Gram-negative organisms, again, just to kind of show, right, we can have, more detailed, things that we can do with them, just like the Gram-positive cells. So again, we want to be able to differentiate if they're cocci or if they're bacilli, and then kind of go through the same thing, to know, the aerobic, anaerobic, and so on. And so here we have other tests, like the oxidase, test that can be done, right, and followed up by glucose fermentation, and we can break things down further into, the different organisms. And so again, these dichotomous, keys are very useful in terms of being able to identify organisms. And so, the slide over here just kind of shows just a quick snapshot, and this is not extensive at all, of what some of these testing situations look like. So, things are very heavily automated these days. And you can, screen for large numbers of tests at the same time. And so, these testing strips in some cases are organized in such a way that you can actually, if you have an inoculating loop with the organisms, you can pull them through the whole panel, okay, at the same time. So essentially, you're inoculating all of the test strips at the same time. And then you incubate, you let the reactions take place. And the results are automatically fed into this, electronic system, and you get a readout that gives you all the information that you need to know about the tests. And so these are very, very useful in terms of being able to do large screens and include all the things that we mentioned in the dichotomous keys over there. So when you're testing for for things like oxidase, for coagulase, you're testing for fermentation of, glucose or mannitol, I mean, you name it, everything, is in there, and you get that readout, and you can quickly tell what organisms, you're actually working with, especially if you're trying to do a diagnosis, you can very quickly determine what you may have, within the specimen or the sample that you're testing. And so, things are done in this way in combination with, the metabolic profiles as well as with, metabolic assays. All of them are used, to provide this information. And so the only thing I want to have us kind of get from this slide, so we have the human microbiome project, it's been around, the different aspects of it are ongoing, and, we have all these different organisms, that have been identified as a result of it in the catalog. And so the whole point of, of this, that we want to kind of get from this slide is that information that's obtained, from these, and some of them, the numbers have gone quite, gone much higher than what we have over here. But the last point is the key. So the idea is to get information on the interactions that we have, between and amongst organisms that we have, in the microbiome, and also between interactions we have, in healthy states and in disease states. And so, if you don't know what you're looking for, sometimes, you don't know which direction to take. But if you have an idea of what organisms may be present, then it makes a difference, in terms of how you approach, the prevention and the treatment and things of that nature. And so this, particular slide over here, just showing the distribution that we have, and we mentioned this last time, of the different organisms that we have in different areas of the body. We want to kind of just focus, there on the mouth and oropharynx area and the GI tract. Okay, so we see the distribution of the gram-positive and gram-negative organisms. And the reason why we're including the GI tract is that, we mentioned the fact that something like Helicobacter pylori, that is not an oral microbe, in some situations is isolated from the mouth. And so the idea is, okay, how is it getting up there? Is it contamination? Is it, that it actually can be there? And, how do we explain that? And so, knowing these interactions and knowing what we have, in the particular locations normally helps when you have an aberrant situation like this one, where you have Helicobacter pylori, showing up in the oral cavity when you would not normally expect it to be there. So again, look at the table, but focus right there within the oral cavity and the GI tract organisms. And so before we get into the detail that we're going to do from beginning tomorrow and into Monday and Tuesday of next week to look at the different types of diseases, we want to kind of still have an idea of how it is that infection occurs. So we talked about Koch's postulates. Everybody kind of remember that. We want to remember what those postulates are, and we also want to remember, why in certain situations, the whole set of postulates may not work, right? But they're still very useful. So, in addition to that, okay, we're looking at the things that happen when we have a chain of infection going on. So we need to have an etiological agent, okay? We always want that. Sometimes it's not as simple and clear cut as, okay, this one organism, therefore we have this infection, right? And so, with the diseases of the oral cavity, we're going to find out that that's the case. We have, polymicrobial infections. So we need to have an agent identity. We need to have some sense of virulence of that agent. We need to know the dose, inoculum, how much, of that agent is present. How did we get exposed to it? Was the transmission for getting that organism there? And then susceptibility of the host to the agent. And so, in this last point, we're more thinking in terms of the immune status of the individual. So if people are immunocompromised, if they have chronic conditions, right, then, they can be predisposed to things that may not affect an immune competent individual. So that last point, is important. So in situations where we have an immune competent individual, the dose, the inoculum might be so heavy that even if the immune system is okay, just by sheer numbers, we're going to get an infection going on. So again, we want to remember, so we have this interlocking situation where we have the agent, we have the virulence, exposures, and dose, etc., all together. And if we have an infectious process ongoing, right, the pathogen, must contact the host, it needs to be able to survive, okay, for it to cause the disease. If it survives, right, it needs, suitable environment, etc., nutrient source, and so on, be able to protect it from other organisms, within the environment or from products of the host. And if we have virulence factors, we will be able to invade. And so in this case, we can either resist the host immune response, or we can, outcompete commensals that are in the area. So when we look at this, we're looking at the organisms that we're going to encounter in the oral cavity, and we've already mentioned Porphyromonas gingivalis. So the question is, at what point does an organism like Porphyromonas gingivalis take over? Okay, to where we have, large amounts of the cells growing, okay, impact the biofilms that we have, and actually lead to, the inflammatory activity that's going to occur and recruitment of cells, okay? So when we look at this kind of a thing, that's what we're kind of thinking about, okay? So, we need to be able to have, where the organism's coming from, right? So if we have Porphyromonas, we have Treponema denticola, things that are not symbionts, where are they coming from? Okay, so how do we get them into the oral cavity to begin with? Okay, so that's an important thing to keep in mind. And so, we get them in, so the different things we talked about, right? So we can, have ways to survive within the host. So we're producing enzymes, proteases, things, metallic proteases, things that can destroy the tissue and allow for invasion to take place. Toxins, can be produced, other invasives, besides, the metallic proteases. And so those things allow the organisms to take hold and get established. And so, as we get more cells into the environment, we begin to establish and kind of firm up the biofilm, okay, to make sure that these organisms, can survive. And then, we have nutrient acquisition, etc. Some of the organisms might actually, get into cells, okay, so they might actually invade, the host cells themselves. And so if we now have at the same time an immune response generated, okay, so both innate and things progress up to adaptive immune response, we might begin to have IgA production, we might be able to have, components of innate immune responses that will kill, we might be able to get in, neutrophils, that will kill, so we can have cells die. Okay, but the thing is, we're not clearing everything, right? So, we still have enough cells available that are still responsible for the pathogenic state that we're going to find, okay? And so this kind of helps us, in a way to think about the processes that are involved. But then, if we have these biofilms established and we have, damage from the cells, and as well as the immune system, trying to get rid of the organism, participate in furthering the pathogenesis, then the thing is, how do we get rid of these organisms? And so with virulence, we can have, a number of things occur. So organisms, will produce, toxins, they may produce, proteases, that can damage, the mucosa, allow things to invade even towards the lamina propria of that, of that mucosal lining. Any of those things can occur. So the virulence factors are important in terms of determining the degree to which the pathogen can cause damage, invade tissue, and increase infectivity. And so, the host, again, the ability of that pathogen to survive, outside of that host is pretty key. So we're going to look at, some of these virulence factors we're going to pull out some main ones that we want to know. Okay, there are several different ones. We don't have to know every single one. You know, down the road, if somebody is interested, you know, you can easily look them up. But we want to kind of know the, you know, the major ones that concern us, you know, for the discussion here. And so we can have what are referred to as pathogenicity islands. Has anybody heard that term before? Pathogenicity islands. Okay? And so these, you know, again, they're important because, you know, the virulence that we're talking about, you know, can be contributed by these things. And so we have, you know, portions either on the bacterial DNA itself or on the plasmids in the bacteria. Okay, so remember, you know, the plasmids are found in the bacteria, so they can replicate autonomously within the bacteria. They can encode proteins that can impart virulence to the host bacteria. Does that make sense? Okay? Make sense what I just said? Okay. And so within biofilms, we can have exchanges of plasmids amongst bacteria. So which means we can increase virulence. You know, if we have these components that are encoded on the plasmids, and if they're shared and so on, we can increase the virulence of these organisms, okay, by being there. We can also have on the chromosome, the single, nucleoid chromosome of the bacteria, we can have also in there encoded molecules that can impart virulence to the bacteria. Okay, so from the plasmids, from the bacterial DNA, and so we can have this, increased bacterial virulence. And if you have non-pathogenic strains of the same organism or non-pathogenic organisms within the biofilm, they will not have these molecules, right? So only those that are producing these, markers will be the virulent ones, okay? And so if the sequences, that encode these molecules are looked at, right? You can see differences, within those regions, okay, that allow for this, virulence, to occur from the products. And so we can have, ones that encode for things like mobility. We can look at the GC content of the particular sequence in there, and so on. And so we can, again, spread this thing amongst the bacteria that we have within the biofilms. Okay, so the first thing we need to remember is the virulence issue. Okay, so what does virulence mean? Okay, and how can we have this virulence, okay, be present amongst the organisms that, for example, we can have in the biofilm? Okay, and so we need to remember that we can get it from the chromosomal, the bacterial DNA, as well as from the plasmid. Not being mentioned here, but I just hasten to add it so that we kind of have it in the back of our mind. We can also have phage, bacteriophage, move things around as well and increase or not virulence that we have in bacteria. So wherever you have bacteria, okay, in the biofilms, right, we're thinking about their plasmids, we're thinking about their phage. Okay, does that make sense? We can't talk about them without mentioning those two things. Okay? All right. And so we can have them transferred. So we have this table over here, okay, showing, pathogenicity items and the products that are made. And so on this list over here, okay, we have a number of, organisms being mentioned. We're going to single out, okay, a handful that we're going to want to know. Okay? So the first one that we have on the top, E. coli, okay? We want to look at that, okay? We want to look at the Helicobacter pylori that we mentioned, right? And then we want to come down to Salmonella and Shigella and Staphylococcus aureus, okay? We know in passing the whole business about Vibrio cholerae. We'll make mention of it in another couple of meetings, just to emphasize the importance of the toxin and how the toxins work, so we'll see the AB type, toxin situation. And, the other one over here is the Yersinia, that, again, in passing, we're mentioning it right now, because we talked about the iron uptake, at the beginning of the meeting today, and we mentioned the siderophore, with uptake of iron. And so again, the ability to be able to, scavenge and take up the iron, okay, in individuals is, considered a virulence factor, because it makes the infection that much more serious when that's going on.
Can you repeat the ones that you
So we have E. coli, Helicobacter pylori, we have Salmonella enterica, Shigella flexneri, and Staphylococcus aureus.
Okay,
Those ones. Okay? So the other ones, just in passing, just so that we know, because again, like I said, the Vibrio cholera one, that's pretty classic. Okay? The whole business of the organization of the toxin and how we have the AB, toxin, how it works. So we'll see an example of that. So we don't, we're not focused on that right now, but the ones that we mentioned. Okay? So we want to know those. Okay, so one more slide and we'll take a break. All right, so virulence factors, so we're having, adhesion, being able to adhere to surfaces, okay, and colonize those surfaces is also important for pathogenicity. And so the table down below just kind of gives you examples of those. And so we've already mentioned the fimbriae, right, the pili. Everybody kind of remember those things we talked about? They're used for attachment and adherence, okay? And so we need to remember that. But then when you look in here, okay, the ones that kind of involve us, in terms of what we're talking about are important. So we see Streptococcus mutans, okay, in the table, everybody kind of see that. And so we see, with the dental caries, and then, we're using sugars for the adherence, and we're on, the tooth surface, with the salivary glycoproteins. And so that one for sure we want to remember. Okay? All right. So let's take a 5-minute break over here, and then we'll come back and continue. And so we're looking at some of the other things, like if you remember the figure we looked at that shows the interaction between cells establishing environments within the host, and having those the established communities leading to pathogenicity, right? So regardless of what type of infection it is, I suppose we could put it that way, right? We have the portal of entry, like we saw in the figure. So we have, where things come in. There's the question about where are they coming from. Okay, sometimes very directly. Other times, like in the case that we're looking at, the oral cavity is not so, it's not so clear. And so we have different ways of having, organisms come into the body. And then, we have adherence that can occur as we begin to establish infection. And this is true whether it's bacteria or fungi or parasites. There always is that point, where things have to, get established. If you're looking at juveniles, they're penetrating, tissue or getting into cells. That's all part of the process. And so we have adhesins, okay, the different types that are responsible for adherence. And then, we have the colonization that takes place. And so, again, once we're being colonized, either we're going to reproduce, right, divide, and have an increase, in cell population like we can with bacteria. Or, in some of the eukaryotic type infections, we don't do that. We have products that are released that end up, causing damage and so on. So again, colonization, this is where we're going to have the microbial production on or within the host. And, there may be no invasion. If we don't have any virulence factors that permit that, we're just going to find them, in one location. So that is important as well. So what are the things that we need? So we've talked about the adherence structures. the pili, the fimbriae. We can have, additional structures that the cells elaborate, in terms of products themselves. And so when that happens, we're going to have, populations of cells in cases in bacteria. We can have, in addition to the pili, the fimbriae, the cells may also have capsules. We talked about capsules as either carbohydrate-based or protein-based. The most common of the capsules that bacteria will produce, are carbohydrate-based. And they can use this to attach to surfaces, and within the host, the presence of the capsule on the bacteria is an immune evasion tactic. So which means, host cells like macrophages and, neutrophils, things like this are going to have a hard time, phagocytizing cells, with those capsules. And so, we need to kind of be able to distinguish, each of those things. And we can stain for the capsules specially, and we can actually identify them. So again, the key point, as we're going towards, these things like Porphyromonas, is that just because we're colonizing an area does not mean that we're going to damage tissue in that area. So if we don't have the products from the cell, and if we're not eliciting an immune response that may recruit cells, we may not cause, a tissue damage in the location. Then we have the ability to invade. And so again, here, we have, the infectivity, organisms are present, the ability to spread, right? And either the cells we're on or nearby can phagocytize or endocytose and take up cells, or we can have the pathogen able to invade, directly by itself. So, active or passive processes. And so if we're active, we're producing proteases, and amongst these, can be things like, collagenases, hyaluronidases, any things that can get through the, extracellular matrix, things that can get through the epithelium, things that can get to the lamina propria of the epithelium, things of this nature, will be things, that have lytic activity that can promote the movement of cells, to these locations. If we're invading to within cells, then there might be receptors on the surface of cells. If we're not being just passively endocytosed, we might be binding to receptors, and we might actually have, a clathrin-mediated type of receptor-mediated endocytosis taking place to get the pathogen into the cell. And so again, if we can get through the extracellular matrix, if we can get to that basement membrane, and if we can get to, the lamina propria area, then we can be carried into the blood vessels, and we can get to other sites within the body. So again, carbohydrate protein complexes, can be degraded, by products from the cells, and this can allow for invasiveness to take place. So, other things that we consider passive, so again, if we have, spread, through already existing lesions, if we have, insect, bites, we already have, wounds present, and then we can actually now, use that avenue to get deeper into the tissues. And so again, with the oral cavity, when we look at how we get things to become systemic, okay, so that's one of the last things we're going to look at, as we finish, this course. We're going to find out that, having the bacteria present in the mouth, we can get them into the bloodstream, we can move them, into, places like the heart and so on. And so in that case, we can have a more systemic spread of organisms that don't normally, belong, in one location, but now they're there because, we already have them present, in lesions and so on in the mouth, and now they've made their way into the vascular system. And so, here, so we mentioned, some of the enzymes. So we mentioned coagulase, right? I just mentioned collagenase, just now. We have, hyaluronidase, which we also just mentioned just a few minutes ago. But we have some other things, okay? So, these virulence factors that are produced that promote, invasion. And so, when you look at the list over here, right, besides the ones that we've already mentioned, We have, things like protein A, protein G. Now, when we, if you're working in the lab, if you've done any kind of, pull down assays, or if you're doing, precipitations, you probably, no doubt use protein A, for those kind of things. But, the bacteria didn't produce protein A just so we could use them in the lab for those assays, right? They produce them because they're actual, virulence molecules, okay? So they can bind to immunoglobulin. So protein A is the longest, known one. It's been used, for decades by now, going about 40 years or so. But in any case, it can bind to the FC region of immunoglobulin, IgG. And so when it does that, then it prevents, complement fixation from taking place. And so because it binds there, we use it, on the bench to pull down antibodies, okay, that are bound to some antigen, okay? But, the main use of it is here, to evade, immune attack from within the host. And so we have those. And so you want to look at this table, and, if we see any of these, mentioned, we talked about beta hemolysis, everybody remembers that, right? The clear hemolysis on the blood agar flakes. So the hemolysis, when they're produced, they're produced because they can do that. They can, hemolyze the cells. So these are all important virulence molecules that bacteria produce and make them, more pathogenic, when we have infections and so on. So again, please look at this table over here, to be able to see the different things. So another one that I want to mention, so we have the hydrogen peroxide that can be produced and so on. And so we have the organisms, things like mycoplasma and so on. Remember, they don't have the cell wall. So when we talked about the peptidoglycan, you all remember this from last week, we mentioned that we have organisms like that don't have that cell wall. So the normal things that you would do to a regular cell wall and a gram-positive or gram-negative cell would not mean anything to mycoplasma because it just doesn't have it. And so we see that it has, something else that's, and that's important that it could actually use to damage, the mucosa when it's in the host. And so we can damage epithelia, in a variety of locations, and this allows mycoplasma to take hold. So this table is pretty important in terms of, virulence molecules, and if we're going to be invasive, right, then here are examples of molecules that the cells will make. And this can allow them, to breach, that mucosa, right, in places like the GI, get through that, get through some mucosa, get all the way past the basement membrane into the connective tissue area. And so we can now get into blood vessels because we have all those capillaries there, and we can, move from there. So, important to kind of keep track of this. And so once we get bacteria in, and so, we now have access to different organs. So again, the example for us, most directly, when we talk about how the bacteria leave the mouth and enter the blood circulation and end up, in the heart, and we have them around the heart valves and so on, how do we do that? So this is the route we're going to take to get there. And so if we have viable bacteria in the blood, so we have a bacteremia. So if we have, the organisms or the toxins and different things like this, we can talk about a septicemia. Now, different organisms will do different things. Okay, so we have Clostridium. Okay, we have toxins that are produced. Good example is Clostridium tetani, right? So we have the toxins produced. We have tetanus that's going to occur, but the organism itself is not the one causing the problem, but the toxins that are being produced are causing the problems, right? So we don't have an invasive Clostridium situation. We have the toxins being produced that are important. Bacillus anthracis, Yersinia pestis, which we saw on the bottom of that list, with the sideropores and all of that, right? So these are the examples of anthrax, causing organism, plague causing organism. We have, many virulence factors, but they're also invasive, okay? So when we get back into those biofilms and we see Papillomavirus, we're asking ourselves, what is it doing, right? So, is it, you know, an early colonizer, is it a late colonizer, is it intermediate? When it shows up in there, what is it producing, how is it affecting the other organisms that you have in the biofilm? And more importantly, what is it doing to our host immune system? Okay, is it, you know, subverting things so that the immune system is not going after it? What is it doing within that environment? Okay, because again, it's a key organism in there. And so we have different Streptococcus species, right? Everything from the mutants that we've mentioned, you know, with the caries, all the way to pyogenes and, you know, pneumonia, right? Different species of strep, and so they have all manner of virulence factors that they produce, okay? And, you know, amongst the different species, you know, different levels of invasiveness and so on. And so these are things, you know, that we're kind of remembering. So we're looking at the biofilm. We have, you know, this, establishment of the cells as they're growing. And so we may have the result of the biofilm, you know, be the cause for chronic infection, increased virulence, have cells becoming less sensitive to antibiotics. And, you know, so biofilms are notorious for difficulty of getting, you know, antimicrobial agents into the core of the biofilm. So you might kill things around in the periphery and so on, but within the core, you know, there's a lot of difficulty getting, you know, those drugs in there, or whatever agent you're trying to use. And then, you know, because the biofilm is so well established, we can't just have, you know, macrophages and neutrophils and things like that come in and clear the biofilm. That's also difficult because these things are really attached to the surfaces. And so, you know, it takes, you know, physical activity to really remove the biofilms, you know, on surfaces when they're there. And so again, this just shows, you know, the progression, okay, you know, of things. And we're going to look a little bit more closely at what happens. And so we have biofilm formation and establishment within the oral cavity. And so we looked at those first illustrations, you know, at the beginning of the class today, right, showing the tooth surfaces and so on, supra, sublingual, etc. And so now we're kind of wanting to kind of get an idea where are things. And so the streptococci, you know, mutants and others, okay, mutants is a major one, will, you know, stick to the tooth surface. You know, they're utilizing the salivary pellicle that's formed, you know, that's on the surface, you know, of the enamel there. And so the streptococci are thought to be the early colonizers, okay, that you have within these biofilms. And then actinomyces species, you know, will follow. And so they produce this carbohydrate polymers that, you know, kind of firm themselves more on that surface, okay? So you need the substrate, okay? You need to be able to get on there. You need to be able to produce molecules that will, you know, that will enhance, you know, things like capsules and, you know, fimbriae and so on to make them firmly attached. And so, you know, in the case of streptococcus and with caries, as we saw in that first, you know, set of pictures, right? This is important to get that cycle going, okay? For the, you know, be able to demineralize and then do the mineralization again. We need to be firmly established within that environment. And then, you know, we have other organisms that will show up. Lactobacilli, okay? Other non-mutants of strep. Okay, and again, they'll be able to use, you know, things like sucrose, you know, converted to glucan, fructan polymers, will make it the thing more firm. Okay, and so people have looked at this, you know, to understand, you know, who's producing what. Okay, and so again, having these, you know, additional adhesive structures kind of further establishes these organisms on that surface. Now, there are studies that look to see, you know, what the association is between the strep organisms and these other later appearing organisms. And so one of those is the Porphyrin monocytogenes that we've been talking about. And so they produce what are referred to as gingipanes Okay, has anybody heard that term before? Okay, so they're producing that, they utilize those to form these co-aggregates, and then we begin to see interactions with Treponema denticola, and they have the dental license that they're producing, and so you have the synergy that occurs, okay, as we begin to establish this biofilm. And so other studies that people have done, they're looking for other organisms besides, the Treponema that you can also find within the biofilms, and they find that there are strong associations between Candida falciparum and Eubacterium nudatum. Okay, and so you can have more than one organism in this association. And so we need to remember, again, that progression. Okay, so we mentioned Streptococcus mutans. We should be thinking of the caries, dental caries. We should be thinking about the cycle that's occurring, that's leading to that. And then when we think of Porphyromonas gingivalis, we should be thinking of a more progressive thing now, where we're not just on the surface of the tooth, but then now we're in the mucosa, and we're going to have immune system involvement and so on. Okay, so with periodontitis again, we're going to have that. Caries, we're thinking more of the Streptococcus mutans and so on, more aciduric, acidogenic organisms that will lead to the damage of the tooth. We have other organisms, Streptococcus, gordoni, that can bind to sialic acids, on receptors, and that's liver-pellicle. And, so it's not just Streptococcus mutans that we have, so we associate that with the caries, but we also have these other species. And so in terms of the mutants, okay, to differentiate it from gordoni, right, we are synthesizing, these water-soluble glucans, right, and we have these glucosyltransferases that are being produced, and we're making these, polymeric structures that are going to enhance the adhesion. And so again, with all of that, okay, the key term over here is coaggregation, and we begin to now, have this biofilm, fully established. And so we've already mentioned the villi, we already mentioned the fimbriae, but we also have, other proteins. We have the 1,2 family proteins. We have lactose-specific lectin-like, adhesins. The A-like proteins, important for this gram-negative type situations. And then we have, serotype-specific molecules that antibodies, can actually differentiate, okay, that are used for the establishment of these things. So between these two slides, okay, here, okay, and here, we want to remember these organisms. And as we finish, I have a list of them, okay, just to make sure that, we're kind of focused on them, okay? So we have this. So again, we had the term, I mentioned keystone pathogen. So we're going to form the biofilms. We saw kind of a quick way as to how this thing forms. We're going to look at it in more detail. We'll look at the types of diseases. But then, if you have biofilms, we have the, process called quorum sensing. Okay, who's never heard that term before? Quorum sensing, okay? Pretty important when we have biofilms. And so, organisms, within the biofilms essentially can either promote the recruitment of organisms to the biofilm, as well they can repel organisms that, they do not want in that community. Does that make sense? So, one of the ways, that we can have this essential, recruitment and so on, to kind of sense and identify or determine what's present in the environment that's being produced either by other organisms, right? Is this process of quorum sensing, okay? And so the numbers of the cells you have within the biofilm is also regulated, okay? So that you don't have, overgrowth of certain members of the community, in comparison to others. And so this whole process, quorum sensing helps to do that kind of regulation. And so these figures over here essentially, kind of showing you what that process is. We have other ones just showing examples of that. Now, this this figure over here is important for a couple of reasons. So, okay, kind of orient yourself, right? So we have the tooth surface, we have the salivary pellicle, right? And then now we're going to get all these different organisms as part of this thing. And so you see, the various molecules, that are available, right? That will allow for the processes of getting these cells, together. And so you have things like, a protein-rich protein, you have, alpha amylase, etc., silylated, mucins, etc. But then you see the initial establishment of the film, and so we already recognize, some of these organisms there. And if you look all the way, to the top there, we just kind of want to focus on that for now, you see the fusobacterium that's there. And so what we want to get out of this slide is the following. So we have those organisms that are early colonizers of the biofilm. We have the organisms, that are essentially intermediate, and then we have, late arrivals. And then, we have those organisms that essentially form the bridge, okay, between the core and these late organisms that are coming. And one of the organisms that functions in this way is fusobacterium, okay? And so again, with the quorum sensing, right, so we're having these cells communicate, and they're producing molecules that aid in cell recruitment. And so if you're interested in preventing biofilm formation, wanting to understand interaction between members of the communities, then you want to know what the molecules are that are used for cell-to-cell communication. So that if you can prevent, the production of them, you might be able to break down the biofilm and prevent, the coaggregation that occurs. And indeed, in the literature and in the studies, that's exactly what people are doing. Some of the studies involve, for the, having volunteers in the studies, have these chips that are inserted, in the mouth to allow, the films to establish, and then getting those out, and then now being able to see what we saw, what we're looking at over here, who's present, okay? So in the amount of time that the chip was left in the mouth, to allow the colonization to take place, right? In that amount of time, which of these organisms, are now there, right? And which ones are missing, right? And, what molecules can you identify being produced by these organisms that are found in that community that you have just now had occur on the chip that was, that was inserted into the volunteer's mouth. So there's some different ways of doing this. So again, quorum sensing is the cell-to-cell communication system used by bacteria to coordinate bacterial behavior, okay, both at the whole population level, okay, through the use of, the different small molecules that are produced, right? So population level, but don't forget, the population is built up by individual organisms, okay, different types, and so even between, okay, closely associated, organisms, we can have this communication going on. So we need to be able to recognize the term quorum sensing, and so on. And so again, if we have immune system involvement, right, so remember, caries, we don't have that, peritonitis we do, so we have, the immune system alerted to antigens, from these organisms, and so we're responding. And so for now, we'll see this slide again when we look at the different types of, of disease. So right now, we're just looking at this to remind ourselves a couple of things. So when we have the bacteria, right, and, we have the cells within the environment, they have the toll-like receptors, they're going to be able to bind to this, pathogen, associated molecular patterns and so on. And one of those that we already mentioned is the LPS. So everybody kind of remember that? LPS, we do that. And so once we do that, okay, the innate immune response, right, with the toll-like receptors, interacting, with the PAMPs, right, we're going to signal through those cells. Downstream, we're going to have, pro-inflammatory cytokines being produced. And examples of them, IL-1, TNF-alpha, things like this, right? We're going to have those generated, and those will now be responsible for getting additional cells into the environment, so we're going to have inflammation occur. And so when we do this, right, we're essentially now not on the surface of the tooth anymore, but we're on the mucosal surfaces. We're in the gingival sulcus. We're looking now at full-blown infection pathogenesis is going to occur, and the immune system is involved. Okay, and so we're looking at these things, and so to that point. The second point we want to get for now. Okay, so we see what happens. We have macrophage involvement. They're going to come in. So it's recorded over there for lipids, but they'll also phagocytize the organisms as well. And they're going to be secreting, IL-1, TNF-alpha, etc. We're going to have small cell proliferation. We're going to have C-reactive protein, also present, and we're going to begin to have coagulation occur and so on. And so, the involvement of the immune system, plus the bacteria themselves, okay, and the products that they're secreting, combined together, right, to cause the pathogenesis that is seen. And that differentiates it from the situation with caries. Okay? And so we want to see that. Okay. So bacterial adherent structures including villi and fimbriae. Okay? So, these are the things that are going to be used for attachment. And we want to say a few things about endospermatic infections, and we're going to have a whole period associated just to look at this, but we want to differentiate things. So we made a differentiation between, between caries, periodontal disease, and then now we looked at the infections over here. So again, urinary canal infections. We have caries. Sometimes the origin of that, and then we can get the bacteria in there. There's a lot of discussion in the literature as to how to really think about this. So in the way we've talked about caries and periodontal disease, we can actually mention organisms that we know, okay, if we see them, we know this is going to happen, right? And we can make associations about the presence of those organisms and the pathogenesis that will result. When we look at the root canal infections and so on, and the bacteria that we find in there, they're not ones that typify. Okay, so these are organisms that can come from other lesions, other places, and they're now, if you will, contaminated. Okay, the area, but they're still bacteria, and we still have to do something about them. Okay, so here we almost have to treat things the way you would do when you have wounds. Right, so if you have an insult to the skin, you have a wound, you have bacteria that's there. So let's say you had a puncture, wound, whatever, and bacteria was on the item that did the puncture. Now you have the organisms there. And if you have anaerobic organisms, you're very concerned because then now they're all the way in there and we have to clean them up. So you pretty much treat these lesions the way you would do a wound. So you will see the same language being used, right, when you're talking about endodontic infections and lesions. You do debridements, you clean out, you follow up, you check to see if you still have bacteria and so on. And you may not see the same bacteria, in the same lesion if you examine, 2, 3, 4 different people. Does that make sense? And so it's a whole different approach, to think about bacteria in this case. But we're still in the mouth, we still have a lot of bacteria there, so we have to still keep numbers down, okay? And so we can have, extensions of caries as the most common route. Bacteria from saliva, it's associated with periodontal disease already, mechanical exposure, trauma, any of these things can get the organisms, further in. And so the kind of organisms, that are associated, non-hemolytic streptococci, alpha GABA type. So I don't know if you remember that we had the dichotomous key thing over there, and we have the various Lancefield groupings of the organisms, right? So we can find these non-hemolytic strep, and we can find lactobacilli, but we can also find this group right here, right? So when we're looking at this group, right, and if you see Porphyromonas gingivalis over there, Trichoderma denticola, we already know what these are associated with, right? So that's where they're coming from, okay? And so we also, so if you find, Porphyromonas gingivalis, okay, we already know about it, that's where it's coming from, but then we have other species of Porphyromonas, okay? Everybody notice that there's another one there, okay? So we have the endodontalis, species that's there, and that's considered opportunistic, okay? But, if things are pretty serious, we need to be able to differentiate the two, okay? So these, differentiation, and we'll look at this, and I have, I think I have an agar plate, image, that we're going to be able to look at. But we can differentiate them based on, the colony characteristics, how they grow on media, how they look, under the microscope, they're gram negative, we can look at the features of them, so we can tell them apart, okay? And so we have them, and then we have other, organisms that have, actinomyces, a species in here, so again, this is, associated with failed root canal treatment and all of that. And so the organisms, that we find in these type of lesions, they're producing endotoxins, these are gram-negative organisms, proteases, things like collagenase, hyaluronidase, so again, they can do a lot of damage, okay, and so on. And so when we think about the organism, so we already kind of defined opportunistic, commensal, right? We mentioned the symbiomes, we mentioned the pathobiomes, everybody with me how we did all of that. And so, when we have the commensal organisms, they can become pathogenic, okay, if they get into a different environment. So, in the mouth, they're okay. If they get into the blood, they get into some other tissues, right? We can have problems, right? So we can have, pathogenicity occur. So if we have any breaches, okay, of the integrity of some tissues, and we get, cells into the blood, we can move them out of the oral cavity and so on. And so within the new environment, we're going to have them cause, infection, especially, if they have factors that are not more virulent, we can have them cause infections. And this over here is just, we're not doing too much of it except just to know that this occurs or this is there. So people who work in this area who are interested in essentially identifying and cataloging and trying to make sense of the consistency of organisms that are found in root canal processes, will produce lists like this. Okay, so in other words, okay, if you were to isolate, okay, organisms, from these processes, and if you plated them out, and if you characterize them, which ones are you going to find, right? So which ones are the most common ones? Which ones are the ones that you least observe? Does that make sense? That's what this list is. And so it's pretty much, looking, according to the phyla and, the particular, classes of organisms that you find in there. And so we see that the high numbers, okay, include the Bacteroides group, Prevotella, Lactobacillus, a lot of the Streptococcus, you will find. And then as we go further down, we see some Clostridium species, Fusobacterium and so on, all the way to down here, Enterococcus. And so it kind of makes sense. So, okay, so Enterobacteria, you don't want to find a whole bunch of those in the mouth. Does that make sense? So Enterobacteria you typically associate with GI. Okay, so if you find, some in there, it's not because they're a normal, member. It could be contamination, and so on, right? And so the whole point of it is that, again, it's good to know, okay, so that if you see something unusual, right, you can, you can explain, okay? And so that's just the basis for this list over here. But nothing, nothing beyond that, just to know that it can actually do And in the minute or so left, we just want to kind of introduce this section, and that is, you can actually use the same techniques that we've talked about, to study these organisms. And so when we're working with bacteria, aseptic technique is pretty important, and it's not a contradiction. If you're working with the bacteria, you only want the bacteria that should be there to be there. Okay, you don't want to have any other thing. And then you have to make decisions about culturing or not culturing, using other techniques, to identify things, and then of course, within the environment, you maintain the sterility. So just real quick, over here on the overhead thing, let's see if this will work. Okay, so we mentioned some organisms, right? And you had those tables, right? Remember the ones we showed twice that had the list of gram-positive organisms, the list of gram-negative organisms. Everybody remember those tables? And we said we want to make sure we want to look at them, okay? So we've mentioned, some organisms, right? So Streptococcus mutans, right? We talked about. Okay, Puffermonas gingivalis, okay? And the Streptococcus gordoni, you want to know. And then the Agrigibacter, this very long name, just kind of slow it down. Actinomycetem comitans, this very long name, right? That we see associated with, Puffermonas as well. And then in the diagram, we talked about the interacting organism, right? The Fusobacterium nucleatum, the one that kind of bridges, the members within the biofilm. And then there are a couple of other Streptococcus species, Intermedius and Cristatus, that are also found associating, with Puffermonas within the biofilm. So the one I don't have on here, we mentioned Triponema denticola, okay? So let me just put it just so we have it. Okay? So we're just trying to keep track of where we're finding these things, okay? And so again, the introduction today generally just to get the difference between what's happening during caries, right? The more diverse the organism profile, right? Unhealthy state. The less diverse, healthy state. Okay? Keystone pathogens, more with the unhealthy state, less, okay, with the healthy state, right? Those kind of things, right? And then remembering, the different locations. So we looked at, where we finding the organisms, right? Everything from the mucosal surface, on the cheeks, tongue, right? We're finding, supragingival, subgingival, okay, within the pits and fissures on the surface of the teeth and everything, right? And so on. Then that first slide, we looked at, it has a whole list. So please go back and look at that, and you just use the illustrations to kind of help you, kind of remember where things are. But we need to know the names of the organisms. We need to know their gram-staining reactions. We need to know what makes them, if they're, pathogenic, what are they producing. So we have the table that lists, the various proteases, collagenase, etc., letting us know that those things are needed, for invasiveness and all of that. And so when we come back tomorrow, right, we're going to begin to look at, first of all, try and make sure we understand how exotoxins differ from endotoxins, right? And then, we're going to continue our discussion to help us look at the diseases in more detail Monday and Tuesday of next week, okay? All right, any questions? And I realized those 3 slides, I forgot to send them. Okay, I'll do that. On my way coming over here, I just realized I didn't do it. But I'll make sure I get it to you. Okay, any questions? Okay, we kind of know how you want to treat those tables? Yeah.
Did you say endotoxins were produced more by
Gram-negative. Yeah. And so that's the whole lipid, core polysaccharide, O antigen, that whole thing, right? We want to kind of remember what that looks like, right? And so also we want to remember, why the lipid A is so, dangerous, to have, right? So the gram-positive organisms, when we're talking about them, we're pretty much talking about exotoxins, of which we've seen one example with the hemolysis, right? On the blood agar plate. Yeah. Okay. Other questions? Okay. All right, so we'll come back, tomorrow and, continue.